Hello everyone and welcome back to Creation Myths. I have kind of a big one for you today. Today's creation myth is the idea, the concept, the claim that there are separately created kinds that we can identify. Now, before we get into any of the specifics here, I have to point out kind of a funny coincidence. Not that long ago, as I sit here recording this, friend of the channel, Gutsick Gibbon, Erica, released her own video on created kinds and the problems with that concept. Now, Erica and I have collaborated a whole lot in the past on a bunch of different things, but this is purely coincidental. We didn't collaborate, we didn't plan anything together. We just happened to be working on the same thing at the same time, unknowingly. So before we go any further, I want to direct everybody to also check out Erica's video on this topic. It is, as you might expect, longer and more comprehensive than this one. She goes into a lot of details about like the history of the concept of kinds and why it exists in the first place. Things that I'm just going to kind of breeze over. So if you want the more comprehensive version of this point, go check out Erica's video. Everybody should do that. It's linked down below. So that being said, what are we talking about? What in the world are these created kinds that we're dealing with today? Well, the underlying idea here is basically there are two causes for this. One is that creationists can't deny that speciation is a thing that happens. You can't deny the fact that new species appear, right? We have way too many direct observations, fossil examples of this. We can't deny that that's a thing that happens. So creationists are forced to incorporate that into their young Earth model. We also have way too many individual species for Noah's Ark, even considering you're only dealing with like terrestrial vertebrates. Even then, way too many species to fit on the Ark. So the solution to these two problems that creationists deal with is to say, okay, we don't have every species created separately. That's like the old idea. We don't do that anymore. Instead, we're going to have created kinds, right? We're going to have common ancestors for groups of species that are similar to each other. And this figure from Answers in Genesis really does a good job kind of visualizing this concept. So you have at creation kind of your ape kind, your dog kind, for example, and it's going to diversify. But then you have the flood and that wipes out all that diversity, right? So now you have what are called your ark kinds, right? Your common ancestors that get off the ark. And then once you have, the flood is over, the floodwaters recede, you have rapid post-flood speciation, right? So everybody in this dog kind right here is actually related through common ancestry going back to that ark pair, right? So in other words, kinds are things that actually share common ancestry with each other, and different kinds don't share common ancestry. They were created separately. So there are two parts to the creationist argument that created kinds are a thing. One is that kinds are real and represent separately created groups of organisms which do not share common ancestry with each other. And that last part's really important, right? You've got your dog kind, your cat kind, your ape kind, your this, your that. Within the kinds, they all share a common ancestor. But across the different kinds, they were actually created separately and don't share common ancestry. The second part to this claim, and a really important part for what we're doing here today to show why this idea of kinds is wrong, is the claim that there are actual detectable boundaries between kinds that we can use to identify where the lines are and delineate what the different kinds would have been at creation or immediately after the flood, right? And it's not just that kinds exist, it's that we can actually figure out what they are. That's a major part of this argument for creationists. And there are a lot of creationists that are engaged in this project of trying to figure out where those boundaries are for the different kinds. So in order to make sense of this, in order to show why these claims are wrong, we have to go through a bit of background information in terms of things like phylogenetics and a couple of other topics. So bear with me for a few minutes as we just cover a bit of this background, and then we'll cycle back around. We'll get back to talking about kinds specifically once we have some foundation we can build on top of. So the first thing we'll talk about is phylogenetics, which just refers to the evolutionary history of a group of organisms. And you've probably seen something like this, a phylogenetic tree that shows, in this example, species A through F, and how they're related to each other through their pattern of common ancestry. So every branch point on this tree is called a node, and the nodes represent common ancestors of the descendant group. So like B and C share this common ancestor right here, A, B, and C share this one right here, and they all share this one right here at the root of the tree. When we are classifying organisms according to their evolutionary history, we are doing this thing called cladistics. Cladistics is a taxonomic system based on phylogenetics. So we're going to use what we can figure out about evolutionary histories to determine things like species and genus and so on up the line, all the way to kingdom and domain. Now, cladistics is built around these groups called clades, and a clade is what we call a monophyletic group. This is a group that contains a common ancestor and all of its descendants. So in red right here, we have a clade. This is a monophyletic group containing E and F and their common ancestor at this node right here. Okay, so that's a clade, common ancestor and all of those descendants. Cladistics, the field of cladistics, or the practice of cladistics, is based on clades, hence the name. It's built around monophyletic groups, because the way this works is you build nested hierarchies. You build groups within groups, and each larger group is a higher taxonomic rank. Okay, so looking at this phylogeny right here, right, we have this group in red. Well, we can put it within this clade in blue, which we can contain within this clade in pink. Okay, so we have this box in a box system. This is a nested hierarchy. This is how we do taxonomy, which is just naming and classifying things with an evolutionary foundation, right? And again, we call this cladistics. We can apply a specific example to this, right? So let's say in red here, we have the genus Canis, right? So like dogs and wolves. That group is contained within family Canidae. And Canidae is contained within order Carnivora. Okay, so it's a nested hierarchy where each big group, in this case an order, contains one or more smaller groups, in this case a family. Each family can contain one genus, or it can contain several genera. Each genus can contain one or more species. So it's a nested hierarchy built on evolutionary relationships. And as you go from species to genus and so on up the hierarchy, your common ancestors are in the more and more distant past. Okay, so that's how phylogenetics and cladistics works. And this is going to be really important for figuring out the kinds in a few minutes. So let's apply this specifically to kinds, right? Now remember, a kind, using our language that we just established, can be defined as a monophyletic group or a clade that does not share common ancestry with other such groups, right? So each kind is going to be separately created and have separate and independent evolutionary history from every other kind. So if we look at our tree right here, we can maybe identify two kinds, right? We have the one in blue and we have the one in red. If those are actual distinct kinds, then we need to remove their common ancestry because they, in fact, do not share common ancestry if they are separately created kinds. So now we have two separate groups here that share common ancestry within the group, right? You've got A, B, and C have a common ancestor, D, E, and F have a common ancestor, but like A and D do not because they exist in different kinds. So like an example of this, we could say this is Canidae, right? The dog family. And then over here, maybe this is Felidae, right? The cat family. And it's not a universal thing among young Earth creationists, but they'll often put the kind boundaries approximately at the family level. So like Canidae and Felidae are two examples you might use to approximate what a created kind is. So that's why we're using those as hypothetical examples. So why are creationists wrong about the fact that there are created kinds? Because they're not. There's universal common ancestry, right? All cellular life shares a common ancestor. That's the actual fact of it. So creationists are wrong when they say we have these separately created kinds. The first thing we need to do in order to evaluate this claim is to be able to evaluate what kinds are. How can we draw the lines between kinds? Well, to do this, we have to use genetics. And like I said, there are a lot of creationists that are trying to solve this problem, but it's a really, really hard problem. And the reason you have to use genetics rather than morphology, even though a lot of creationists will use different kind of morphology-based techniques to do it, is that morphology is too variable, and it will lead you astray if you try to do kinds based on morphology. And I'll just give you two reasons for that right here. The first is convergent evolution, or just convergence. This is where you have two or more things that end up morphologically looking very similar to each other, even though they don't share a common ancestor with those morphological similarities. And an example of this that I really love are the raptors, right? So this is a phylogeny of just some of the birds, and highlighted in red up here, we have the hawks and the eagles and their close relatives. And they're all very similar to each other, and they're closely related to the vultures, as you would expect, right? They're all closely related. Except down here, we have the falcons. Now, superficially, right, morphologically, you're going to put the falcons up here with the hawks and the eagles. There are distinctions depending on how what factors you use, which is a whole other problem using morphology. There's no standard for what characteristics you use and which ones should be weighted more or less. Putting that aside, in general, you would generally, morphologically, put the falcons up here with the hawks and the eagles. But genetically, we know that they're more closely related to the parrots down here. So morphology is going to lead you astray, in part because of convergence. Another thing that can happen that can confuse you when you're looking at morphology is called plasticity, or phenotypic plasticity. This is where you get very different phenotypes from the exact same genome. You could have two individuals who are genetically identical, and yet they exhibit very different phenotypes, physical traits. And so this figures from a 2010 paper shows some of these cases. So these two individuals, again, genetically identical, but you're looking at very different morphology. And then here's an example with plants, not relevant for the arc, but plants tend to exhibit phenotypic plasticity as well. So this is also going to cause a problem when you're trying to delineate your kinds based on morphology. Another reason you have to use genetics to do it. So how do we tell kinds apart? There is actually a clear, objective, genetics-based approach to being able to delineate your kinds. But to understand it, we need to establish even more background information. So again, we're going to do a little bit of background, then we're going to come back around and put all of this together to talk about why this concept of kinds doesn't work. So the first concept we need to talk about, just briefly, is natural selection. Natural selection, it's a mechanism of evolution, one of many. In general, in a very broad sense, we can define natural selection as non-random changes in allele frequencies within a population. In general, because of natural selection, alleles that are good, they're beneficial to fitness, they're going to increase in frequency over time, and bad alleles, alleles that hurt fitness, are going to decrease in frequency over time. And here's a simple example from a website I really like. This is from evolution.berkeley.edu. They have a really great kind of introduction to evolution thing there, and I encourage everybody to check that out. This is a very simple representation of natural selection. So you've got a bird population that eats a beetle population, and the birds are better able to see the green ones, so they preferentially eat the green ones. So over time, that population that's green with a little bit of brown becomes kind of a mix of green and brown, and then eventually mostly brown. It's because the alleles for brown were more beneficial, so they increase in frequency at the expense of the alleles for green. That's how natural selection works. Now, there's a specific type of natural selection, or a specific dynamic of natural selection called purifying selection. This is when you have a population that has a trait that is quite good. It's well adapted for its environment. It's close to optimal. And when you have that situation, you're going to see selection against new variants of that trait, because you're already about as good as you're going to get for your particular environment, so any deviation is bad. So we can show this graphically here. So what's going on here is we have fitness on the y-axis, some very low fitness to very high fitness, and then variation across the x-axis. And this curve represents the variation that's present in the population. The higher and more blue it is, that's high fitness. And on the sides where it gets lower and more purple and then red, that's lower fitness. So that indicates a suboptimal genotype or a suboptimal allele in the individuals that are down here. Okay, so if we have most of the population that's up here, right, most of the individuals are pretty good, any deviation from that is likely to make you worse. You have a mutation and you're already really good, then that mutation is likely just by chance to make you less good than you were. That means that natural selection is going to work against those mutations that are occurring, and specifically its purifying selection is going to kind of push that population back up towards the peak of that fitness curve, right? You're going to be, everyone's pretty well adapted, you have mutations, those get selected out of the population, and the population stays close to that peak. Now, to be clear, when I say selected out, that doesn't mean an individual with a mutation just automatically dies or something. What it means is they might die, or they're just less likely to have kids, right? They're less likely to survive to reproductive age. Whatever the case is, on net, you're more likely to reproduce successfully if you're up here compared to being somewhere else. That's what we mean by selected against or selected out of the population. The net effect of purifying selection is that the DNA sequence experiencing the selection basically stays the same over time. Mutations don't accumulate because mutations are selected out of the population. Another way to say this is that sequences that are experiencing purifying selection are called constrained sequences. That means you don't have kind of evolutionary flexibility in that sequence. They're basically going to stay the same rather than accumulate mutations and variation over time. The opposite of a constrained sequence is an unconstrained sequence. That's just part of the genome that is not subject to purifying selection. And I want to be very clear. We're not arguing about function here, whether how much of the genome is functional and blah, 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 blah. Doesn't matter for what we're talking about here. All we're talking about here is whether a region is subject to purifying selection or not. When it's not, we call that an unconstrained sequence. And like for most eukaryotic genomes, that's going to be a lot of the genome. So for example, for humans, this is a pie chart with a breakdown of the different parts of the human genome. Most of those components are going to be unconstrained. Things like retrotransposons, endogenous retroviruses, pseudogenes. These are regions that are not operating under purifying selection, and they make up a majority of our genome. So if you look at this pie chart here, you've got your lines, your signs, LTR retrotransposons, DNA transposons, most of what's going on here, and introns, and a big chunk of all this other stuff too, is unconstrained. We can document directly that it's not experiencing purifying selection. And that's the case for, humans, mammals, vertebrates, right? Animals. A lot of eukaryotes have a lot of unconstrained DNA in their genome. And again, this is going to be really important in a few minutes when I promise we're going to get back around to talking about kinds. So the one last thing we need to cover before we get there is talking about mutations. And here's the big idea. Mutations are random. Now, I don't like the word random. You may have heard me say this before because random gives people the idea that every mutation is equally likely. And that's not the case. Some mutations are more likely than others, and some regions of the genome are more likely to mutate than others. So another way you might think about mutations, two words that I like are probabilistic and stochastic. Basically, mutations occur according to a probability distribution. Mutations are not deterministic, and organisms cannot pick which specific mutations happen in response to something happening in the environment. And we've known this is the case since the 1942 Luria-Delbruck fluctuation test. I'm going to take a minute to go through this just so we're crystal clear on what we're talking about here. So this was a test of competing hypotheses of induced mutations versus random mutations. And the way they tested this is they took multiple bacterial populations and exposed them to viruses, bacteriophages. And if you have induced mutations in response to the presence of the viruses, mutations that result in resistance, then across different tests, across different bacterial populations, you should see approximately the same rate of resistance, right? And that's what this left-hand side shows, right? This is what we expect if mutations are induced. You're going to have approximately the same frequency across your different tests. But if mutations are spontaneous, they're random, they're not induced by the environment, then you're going to see a large variation in the level of resistance in your populations. Some populations are going to have a mutation occur early in their growth that confers resistance. So you'll have a lot of resistant individuals there. Some will experience a mutation later or not at all, and you'll have very little resistance, if any. And some will be somewhere in the middle. So they did this test, and what they found was the second thing. You had tons of variation in the amount of resistance to the viruses, indicating that the mutations were spontaneous rather than induced. And that's really important. Mutations are random, they are not deterministic. The big takeaway that we're going to apply is that random mutations can accumulate in unconstrained genome regions. So if you have two lineages that are separate from each other, you can look at their unconstrained regions and you'll see mutations that are different. Those mutations occurred since they diverged from each other, right? Lineage A got one mutation, lineage B experienced a different random mutation in that unconstrained part of their genome. Okay, that's really important. That's the big takeaway right here. So let's bring this back to kinds. How do we determine kinds? We're going to put all of what we just talked about together. The way to determine kinds is to construct phylogenies using unconstrained regions of the genome. And I'm going to spell this out. There's going to be a lot of words on this slide, and I apologize because I like pictures instead of words, but I want to make this crystal clear. So we're going to spell this out with words, then we're going to do it with pictures. We're going to cover all our bases. So even if you start at creation, let's say the different kinds were created with identical sequences in unconstrained regions, even if you start identical to each other, those unconstrained regions are going to experience and accumulate mutations that are uncorrelated across the separately created kinds. Now, in the evolutionary model, right, where we have common ancestry for all of cellular life, all of those differences that we see in those unconstrained regions, they're the product of millions of years of evolutionary history. And different things like humans and chimps and gorillas share a lot of that evolutionary history. But in the creation model, all that variation that we see is compressed into the approximately 4,000 years since the flood. So you have different kinds got off the ark, and you have those unconstrained regions in their genome. Even if we assume that they start off identical in those regions, they're going to experience random mutations or probabilistic mutations that are going to cause those unconstrained regions to diverge from each other in ways that are uncorrelated across the separately created kinds. So if you try to build a phylogeny of multiple kinds using your unconstrained regions, you will not resolve clearly defined separate clades for each kind. Instead, they're all going to be mixed up with each other. If you restrict what you're doing to just within a single kind, then you'll get a nice, neat phylogeny because they do actually share evolutionary history. So you'll be able to do your nested hierarchy within the family or whatever level it is that you draw around your kind, right? You'll actually get a nice, neat nested hierarchy. But once you start crossing between the different kinds, that structure is going to get disrupted because across the different kinds, they're going to experience all different mutations that are uncorrelated with each other. So the clades that will resolve within a kind will get disrupted once you start doing comparisons across kinds. We can look at this graphically. So here's the same picture from Answers in Genesis that I showed you earlier. If you look first on the left over here, within each kind, you can do your comparisons within the genomes of all of these different things, and you're going to get a nice, neat nested hierarchy, right? You'll get your dogs and your wolves and your coyotes and so on, as long as they're all in the same kind. That's fine. But as soon as you start to try to compare across kinds, you're going to mess up your pattern, right? You shouldn't, for example, see that primates and rodents are more closely related to each other or more similar to each other than like primates and Ursidae, the bears, for example, right? And it's true that primates and rodents are more closely related than either of them are to bears. So we should see a nested hierarchy there, right, in our evolutionary model. In the creation model, we won't see that nested hierarchy. We won't see a neat structure across the different kinds. In fact, we expect to see some shuffling up, some jumbling between the kinds because, maybe this lineage right here experiences some of the same mutations as this lineage over here. They are, after all, random mutations. So that's going to mean that you're not going to resolve these two neat clades if you were to try to construct a phylogeny using members of both, right? And that's just a consequence of the creation model and unconstrained regions. They're going to accumulate mutations. There's no shared evolutionary history, so all the variation we see, even if you start identical, is going to be due to what happened since they got off the ark. So even if you assume that they start off identical or very similar to each other, that's going to get disrupted as, all of the variation that we see accumulates in just 4,000 years. So here's how we identify kinds. This is the answer to the question. You use genetics, you use phylogenetics and unconstrained sequences. You iteratively construct phylogenies using your unconstrained sequences, starting with very small specific groups and adding lineages to them. So for example, you make a phylogeny of dogs using their unconstrained, maybe use endogenous retroviruses or some other part of their genome, pseudogenes, whatever you want to use, right? And then you add wolves to that, and you use dogs and wolves. And that should make a nice, neat thing where you've got dogs and you've got wolves. And then you add coyotes to that. You should get dogs and wolves and then coyotes. And it should make a nice, neat, nested hierarchy. You keep doing this. Keep adding groups. Add foxes next. See what happens. If you resolve a consistent nested hierarchy as you add each new group, you're operating within a single kind, right? So you've got dogs and then wolves and then coyotes and then foxes. If you keep that pattern as you add new groups, you're operating within the same kind. But if you generate a completely different phylogeny, if you disrupt the clades that you previously made, and they're now intermixed with new things that you're putting on your tree, now you're dealing with two or more kinds, right? And again, we're only comparing those unconstrained sequences, right? So the idea here is you're going to get neat hierarchies within groups that share an actual evolutionary history, but you will not see that same correlation across groups that do not share an evolutionary history. So once you start trying to construct your phylogeny with members of two or more kinds, again, using only their unconstrained genome sequences, you're going to see a breakdown of that nested hierarchical pattern, and that's how you know whether two things are the same kind or not, right? You have a nice, neat clade, you add something to it, it disrupts your structure, that thing that you added is a different kind, right? That's how you do it. And again, you have to use genetics for this because morphology is unreliable. This is the answer to the question of how you delineate kinds. You're welcome, creationists. I just solved one of your problems. Except I didn't really solve one of your problems because when you do this, what we find is that kinds are fake. There aren't actually separately created kinds. As I just talked about, we can determine exactly what each kind should look like. Phylogenetically, we know how to identify the boundaries. Where do we find these boundaries? We don't. It turns out you can actually do the phylogenetics on unconstrained sequences. People have done it. And when you do that, you get consistent nested hierarchies from top to bottom. You don't see a breakpoint where it stops working. And here are just two examples. These are two papers using endogenous retroviruses. They're both from 1999, coincidentally. So we have constructing primate phylogenies from ancient retrovirus sequences. So you can reconstruct the human, chimp, gorilla, orangutan, and everything else within primates. You could reconstruct the primate phylogeny using endogenous retroviruses. Now, according to creationists, primates are more than one kind. So that should not be possible given that endogenous retroviruses are unconstrained. It gets even worse, though, because you could go broader than that. So in the second one, this figure right here, we have a nice, neat clade for the simians, and we have another nice, neat clade right here for the rodents, again, using endogenous retroviruses. There's no reason for the rodents to all fall out right here and the simians to all fall out right here, considering you're going to have multiple kinds in each. This pattern should not exist, according to the creation model. They should be intermixed with each other and with other vertebrate groups. But instead, you find rodents and simians falling out fairly close to each other, just like you do when you look at, for example, protein coding sequences. You know what predicts this? Evolution predicts this pattern. This is exactly what you would expect if these unconstrained sequences and the variation that we find within them are the products of millions of years of evolution, much of which is shared among the different groups that we're talking about. Let's be a little more specific about the predictions that the two different models make with regard to sequence similarity, nested hierarchies, and kinds. So within separate creation, looking at the separate creation model, we can look within created kinds and between kinds because we expect different patterns. For the common ancestry model, we only have to make one set of predictions because everything shares common ancestry. Now first, looking at constrained sequences, we're just going to say everybody expects the same thing. That's fine, nested hierarchies. I think there's a little bit of, kind of shenanigans going on because in the creation model, this is basically a common design argument, which I've got a whole other video on that. It doesn't work, but that's what's going on here. You're saying, well, common design, things that are more similar sequences. And you know what? For today, that's fine. I don't care. That's fine. But what we really care about are the unconstrained sequences. Right now, we talked about this already. Within kinds, we expect to find nice, neat, nested hierarchies in those sequences. But if we compare between kinds, we should not get that same consistent pattern. And of course, under common ancestry, we predict nested hierarchies top to bottom, not just within the kinds, but across everything. And what do we find? That's what we find. We find nested hierarchies in unconstrained sequences. And just a moment ago, I showed you two papers that are examples of that finding. So when we're talking about the practice of doing science, right, hypothesis testing, what we're doing is making and testing specific predictions. And here we go. Our predictions for separate creation are just wrong. And the predictions for universal common ancestry for cellular life are spot on. Now, there's one more thing to talk about before we wrap this up, and it's orphan genes. Orphan genes are genes that are what we say, they're taxonomically restricted. So they're found in just a single species or genus or some other group. And creationists will sometimes point to orphan genes and say, well, we could use the orphan genes to delineate the kinds, right? We could identify the kinds based on the genes that they have that nobody else has. But nope, you can't do that. That doesn't work, and here's why. First, orphan genes exist at all levels from species on up. So you could find species that have orphan genes, but creationists generally reject the idea that each species is an individually created kind. But you find orphan genes at the genus level, at the family level, at the order level. So where do you draw the line? Which orphan genes count and which ones don't? It's arbitrary. The other problem with orphan genes is that they're under selection. So you can't use them in the way we just did with unconstrained sequences because they are constrained. They're actually experiencing purifying selection. So comparisons across different kinds, you could basically explain with, common design, whatever. It's not an objective standard that allows you to make comparisons. And then the final problem with orphan genes is that they're actually not orphans. Turns out we know how orphan genes evolved. And again, I have a video on this. I'll link it down below. But the problem for today is that even if you identify, say, a family that has a specific orphan gene, you're going to find that sequence in a similar family, in a different kind. So then you can't use that sequence to identify the kinds. And the reason is because we know how orphan genes evolve. They evolve from non-coding regions that acquire the components needed to become protein coding. So, for example, you can look in humans and find protein encoding genes that chimps and gorillas don't have, but they'll often have regions that match the same sequence, except it'll either be transcribed to RNA and not yet translated to a protein, or it won't even necessarily be transcribed. But the sequence is still there in their DNA. So even though the gene as a protein coding gene might be an orphan, the DNA sequence actually shows an evolutionary history in closely related species, even though, according to creationists, those species are going to be a different kind. This figure shows basically that process that I just described. So you have here an orphan gene in this purple lineage, but up here you have closely related groups that have the non-coding sequence that's the same, right? It's the sequences there in their DNA, it just doesn't code for a protein yet. So it's not really an orphan, and it does show evolutionary history, But the bigger problem for using it to identify kinds is that this thing that's supposed to be isolated to one kind is actually shared across many kinds. So it doesn't work as a standard. The paper for this, by the way, will be linked down below, just like all the other papers I have referenced here. So let's summarize what we talked about today. The creationist claim here has two parts. The first is that kinds, separately created groups of organisms, are real. And the second component is that we can figure out the boundaries. There's some way to actually figure out where the lines are between these kinds. It turns out creationists are sort of correct about number two, in that there is a way we can do that, but when we do it, we show that kinds are actually fake, and there's actually only one kind of cellular life. It's all cellular life that shares universal common ancestry, which is probably not the result creationists would hope for when you apply the test to delineate between the kinds. But that's what we find when we do that test. So, separately created kinds as independent biological entities, that is a creation myth. And now you know why. Thank you for watching. I hope you enjoyed and found this informative. Please leave a comment, hit like, share this around, and if you're not already subscribed, please subscribe to the channel for more stuff like this and some other really good stuff. Thank you for watching, and as always, don't get fooled.